Okay, so today we are going to talk about the first and do the distance between 2 points. Between 2 points, okay? And then the distance from 1 point to a lot of other points to other. Then we can see, okay, so how can we use optimization to search, to help us to find the central node, which if we don't know where this central node is, this optimization function will help us to find the point which the sum of distance to all other points to be smallest. Then we will look at this built-in function like k-means, etc., and the spectrum clustering algorithm in spectrum clustering. And then we will play with real single cell data. And use clustering algorithm to classify, to cluster cells into groups. And then if we have time at the end, we will see, okay, how can we merge multiple data set? Merge single cell RNA-seq data. Okay, that's from a different samples. You want to merge into one object and to compare. Any questions? Okay, let's generate 2 random points. Say we have a so the first point. I like my 2.5. Okay, the B is 2, 1. So I use bottom column vectors to represent data point. So what's the distance between them? So if I plot this scatter, I can see A, the first one, A, the second one. So I can plot the figure and hold on. And then I can plot the second point. It may be difficult to see on, I don't know, on your computer. So here is the first point, and that's the second point in different colors. So what I can do to make it in the center, I can set limit, x limit from say 0 to 3, and set my y limit from 0 to 3. So after I do this, so you can see on my screen, we have a scatter plot for 2 points. And also I show the limit of x-axis and the y-axis, so I can put them in the right place visually. Okay, so what's the distance between them? So I can say A minus B. Okay, so that's A. Now, if I so now A is this. If I think this is A1, I make another point, A2. So I just made it up. So I'm going to plot A2 onto this figure as well. So I can do scatter A2 first coordinate X, A2, 2. Okay. The blue point is my A1, and the orange, my A2, and my B is this one. Now I can combine A and A1, A2 into a matrix. Okay, it's by column. Now I want to compute from A1, the distance from A1 to B, from A2 to B. So we want to compute the two distance. Let me label this. So I can do the text A1, 1, A2, 1 as my A1, the point. Oh, wrong. Yeah, by mistake, I labeled this wrong, but you can ignore it. So I want to compute the distance from A1, A2 to B. So you can compute norm A1 to B, A2 to B separately. You can also compute, see, A to B in this way. We need to compute the norm. Let me think. Yeah, so we need to do the vector norm. So you compute not the whole matrix norm, but by column. Okay, what I did is I put A1 coordinates and A2 coordinates together into a matrix called A And then I operate on A itself to compute all the points in A to B. And I use this function, compute the norm by column, so called vec norm. In this case, I can just use one line of code to compute the 2 points distance to B. Okay, consider B as our center. Okay. Now I want you to generate a lot of points in not only A1, A2, but see A1, A2, A3, A4 to A100. We randomly generate 100 data points and put in, or still put in the matrix called A, and then compute their distance to B. Okay, we will compute this. Let me see, the dimension will be 2 rows, 100. So I just make a 10, so it's easier for me. I just make 30, okay? Not too many, not too few, but just illustrate the points. So I want to scale it a little bit by 3. Yeah, maybe by 0.25. Now I'm going to plot a new scatter plot to show all this, the point I generated. So I'm going to use a new figure, scatter. So A, the first row, and A, the second row. Then we hold on. Okay, I randomly generated 30 data points. I will still put my B in there, scatter. B1, B2. And in this case, I'm going to use a field point. Okay, my B is in that orange point over there. So now it's your job to compute distance between all 30 points randomly generated, their distance to B, and the total was the sum of the distance. I can put a lim, so it looks better. Okay. So your job is to compute all the points, the blue circle, distance to orange point, which is in the small b, the coordinates of the point of big A is in capital A matrix. Online students, if you finished, you can copy your answer to the chat so I know you are done. Well, it depends on the random number you generated. So we may have a different answer, right? So I can try on, so just maybe just show me the source code. How did you use to compute the total distance between all blue points to the orange point? Remember, in the k-means cluster, we try to find a centroid for all the other points belongs to same group and their distance to the centroid, and try to minimize that. So we need to have a quick and effective way to compute the total distance between many points to any given single point. Okay, so Alex gave the correct answer. I hope everybody have this. So Alex, what's your number? Yes, also Brenna also gave me the correct answer. So let me try on my computer, see what's the answer for my example. Copy this. Yeah, I get the same answer with Alex. Okay, now it's your time. Please find another B, and visually you may want to find, okay, where is the B? Just give a guess and try to minimize this. Minimize what? Minimize the total distance from the blue points to a new number. You can make it up. See, the new number is 1, 1, and then you compute and see. And we try this and see what our the best solution. Because right now the orange point B It's just a random number. I made it up, but you can try different numbers. Yeah, because I get the same answer with Alex get from his computer, which indicates this random number generator function is not purely random. It is because that it is based on some kind of algorithm to so we start from the same initial value, we will have the same series of the string, the series of a random number. So that's why this machine generated pseudo random number. Okay, I show you an example where I have a new b. So if my b is 0.5, remember the column vector, 0.52. Okay, I'm going to try this, see whether my sum of this is better. So my original b has the distance 33. Now this is this better or worse? The worst. So this point 0.52 definitely not in the center of here. So that makes sense. It's far away from the center. So I get a worse solution. Our goal is to find a minimize this function, minimize this sum, total sum. So what's your answer? So I can give my answer. Say if I have a b is 0.5 and 2, my answer is my total sum. So let me use my d. So that's my the d is my 43.4668. So that's my try. So I search for b and then I got this. I can try another one. Anyone gave me an answer from your side? So I'm going to move this to 1.2. Answer 39. Okay, something like that. Okay. B is 0.5, 1.2. And then D, my D is 39.793. So let's find the best one. So Alex got 31. Looks like this is the minimalist we can get. Anyone else have a better solution? Okay, so Alicia, what's your B position? 31. 1.5 and 1. So that's the best answer we have right now. Okay, that's good. So for this simple solution, maybe we are already finished. So we can try a couple of times more. And then if we don't have a better solution, that will be our best solution. Anyone else can? Oh, there's one better. Brenna found a better one. So, oh, Brenna, your number is different from us, maybe. Remember, you have a.
Yeah, I have different numbers, so I don't think mine counts. I just wanted you to know I'm here. I'm
So
working
your
on it.
solution is local. It's only for yourself. You have a
Yeah.
private solution. So we
Yeah.
are sharing the same. Okay, so let's just assume this is the best solution we have right now. So for this simple project, we can do this, but we want to let computer to do this for us. I remember also Alex asked a question during the lecture, and how can you set up a framework like a computer to search this solution for you? In this case, it's simple. You only have two parameters. You have a B1, B2 that you guess and which found the best solution. And also you know the range of the solution. It should be around this, in the middle of that. But in a more complex situation, you may have no idea what your result best solution is supposed to be in the high dimensional space, or you don't know the shape of the data. So we need optimization procedure to do this. So now I'm going to show you how to do it. But the center of this is you should have this function, because we know how can we judge whether your random guess B is better or worse. Our judgment is from this sum of vector norm A minus B. Okay, we fix A. A is a given data points. We are not going to change it. We are going to change B. The B, then we have a function to tell us whether this is a good function, good solution or not. So we need a function. I'm going to build a function called the likelihood function, like func or something like that, whatever name it. This will take so this function, remember we can create a function called this like function, and I need input. That's my data. I give input. So my object, so this will be my solution. So the solution is D, okay? So that's my D. And also we need a B, because this function need a B and it need its data. So B is your the solution you want to try. And A is your data. And what's your criteria, say whether A, B is good or not. So that's a D, okay? So now we have the function just like this. Okay, to do optimization, you need a function called found minimum search, okay? Is that easy? Okay, you need the name of this function. So you need this like function. So you see, once I gave this, the suggestion of this is to give me the name of function already defined. and that's this. And also you need initial value. Say I want to start from a 0, 0, whatever. This will search to give me best B. Optimal. I have to refer back to some of the chapters I wrote. So X, yes, so this will give you the solution. Oh, yeah, so let me see. We still have to give A to the function. I'm just trying. I don't know exactly how to write this syntax, but I can try this. If not work, I will modify it. Okay, so we need put a tell the function of this. Not enough. Too many. Okay, so that's my solutions. My best B I can get. Okay, that's my solution. So what you can learn from this, to do this optimization, you need a likelihood function. This likelihood just tell you how good, how bad your solution is. It's also called objective function or penalty function. When you see likelihood function, it's based on a probability framework. So you want to have a solution, see this is best using some likelihood overall. But in our case, this sum of the distance is not likelihood. It's some, I would say it's objective. So we can change the names, but it's just a name. It doesn't matter. We need to give the name of the function using this at the name of function to give a pointer of the function to the search engine. So this search engine is encoded in fmin search, okay, this function. And the next parameter is the initial value. So what's your first guess of your solution? You put a 0, 0, it's okay. You put something else. In this case, it doesn't matter, but in some more complex situation, you would have a better initial value to reduce the search efforts, because you gave us a solution which using educated guess, so you know, okay, this is maybe close to optimal, and the search will take less time, okay, more efficiently. But in this case, it doesn't matter, you put anything, and you will get the same answer very quickly. Next parameter here is optional, and there's some option, so I just leave it out. Without giving this, the function will use all the default settings. See how many steps after search I will stop, what's the criteria of the precision I will stop. There's no improvement of the likelihood, and I will stop. So something like those. And the last one is the parameter I really need to give the function, because the function likelihood function need a parameter A here. Otherwise, it doesn't know what to compute, right? Now you can test using this optimal solution. What's your total sum? Okay, let me see. So if I do this minus opt, is that the same as what you get from your manual search? Yes, from the chat, I remember our best solution was 1.5 and 1. And the D was 30.93. So this optimization actually found the better solution for this, right? Slightly better. So anyone have any other questions about this framework of the search? So you can try this framework for any other problems. So if you can think about, you need likelihood function, you need to define, okay, how can I tell whether my solution is good or not? You need to know to define the parameters, because in this case, I have a B, I know there are 2 parameters I need. And you need to know the data, okay? So that's all you need, and you need to know how to write a likelihood function to compute that D. So I don't think in this case we need optimization, because the problem we have a analytic solution, which you can compute the mean of A and to get the best solution, right? So this should be column-wise. So this should be the best solution. So I will put the best solution and optimized B opt side by side and see. So on your left side is theoretical best solution, and on your right side it's the search found it, right? Can you compute which one gave you the best solution? What's the D for? Which one is better? If I remember correctly, we have 30.559 something. So the optimization function actually gave you the better solution in our case. It's even better than the centroid, the mean compute. Okay, so this problem is simple. So if you think if you have the k-means cluster. So you want to use optimization to achieve the k-means cluster. In that case, you will define, for example, k equals 2, then you need to have a 2-point central node. You want to guess, optimize the position. And you also, yeah, so you need that. The likelihood function will be slightly different because you need 2 points. So your B will have not only 2 numbers, but 4 numbers, right? You need 2. You need B1, B2, you have 2 points. And also you need to compute all the points to B1, all the points to B2, the distance. Then you judge, okay, those points belongs to which group. Then you will compute the 2 global distance from the members of them to this and the sum. So the likelihood function will be more difficult to write, but still. I think it's really, you should give it a try. I can give it a try if you say. If we have enough time, okay? So what do you think? We should move to next topic or just keep going?
I had a quick question, which was, how is it that the mean is not the lowest point, like, or the most optimal point? Is that just because of the presence of outliers in a different axis? Where is it?
I think you are absolutely right. If you have a shape which your mean was biased towards some outlier point, in this simulation, the data, the random generator of this centralized everything around 1.5, right? 1.5. So this effect is not so obvious. But yes, I think you are right. If you have an outlier and your average, well, yeah, so your average will be biased to the outlier. But I still, this difference should be a sampling issue because you don't have enough points to cover all the space here. So your central node is not slightly different. So I don't have a quick right answer to your question.
Yeah, I was just curious because from what we went over in class, it was the same way to arrive to the same point, right? Using the mean and
yeah, the mean definitely should be the theoretical solution for this problem. But again, I believe it's the sampling issues, so you don't have large enough sample to support your estimation of the mean. Okay, I think we can move to k-means. Okay, now we need to generate some points that we just look like we have some clusters in there. So I'm going to clean my workspace. So I will load this process data using example. I can give a couple seconds to wait until you have the same screen here. I want to borrow the coordinates for those cells. So if you can have this interface now and click this export and save data and see, okay, I will export my data into the workspace. What we need is the last one, the S. Okay, S will be n by 3. 3, that means the position in this 3-dimensional space, and n is the number of cells. So we need to borrow those coordinates. That's all we need. So if you finish this, you should have a workspace, have a variable called S. That's embedding the coordinates after embedding. And in 3 dimension and 2,509 cells. Okay. So after you finish this, you can close this interface. Now your job is to plot using scatter plot to show these data points, okay, on your figure. So remember we use scatter plot. You can either play with 3-dimensional or without 3, that will be the 2-dimension. 2-dimension is okay, they still be able to work it out. If you know how to do this, paste your code into chat so I have some idea and see what's going on. Now what we are doing here is we are embedding the points of the cell. Each point represents a cell. This cell's t-SNE embedding coordinates in 3-dimensional space. We want to have a quick look about this shape of the cloud of the points, and have some idea how many clusters may in this data, right? Now I want you to use cluster, the figure function to visualize the data points. To do this using 2-dimension, then that's the code. I look at the first 2 dimensionality first two dimensions and then plot the cells. Okay, you can also try the combinations of first dimension with 3 or 2 with 3. Okay, you get a different view of the data, but they're still the same set of data, right? Of course, you can use scatter 3 to plot 3-dimensional figure. Okay. So on my screen, I have a 3-dimensional and also 2-dimensional scatter plot for the same set of data. Okay, next, I'm going to show you how to use k-means to cluster those points. I will use built-in function. I'm not going to rewrite the code because it's possible, but it will take me 1 day, maybe. So I will show you the code, and then we take a 5 minutes break and come back, see how can we label those different colors on that figures, okay? So please tell me how many clusters you want to group them into. Select your K, so we need a K. I would say 5. Is that a good? 1, 2, 3, 4, 5. Okay, I'm going to set 5. So what's the input for this? So input for my k-means just S, this position of those cells. And my output is a variable called the membership or category labels of the cells. So, okay. That's it. So let's see what output looks like. Okay, my output is a vector 2,509 rows and just 1 column. So this vector contains integers to label each cell their membership ID. So because I have a K there, so this will be integers from 1, 2, 3, 4, and up to 5, because I will ask for 5 different groups. That's it, okay? So internally, it's doing the one we just told you in Tuesday's class. It's found the random position of these 5 central nodes, and then update the position at the end. They stopped and said, look at the nearest neighborhood of the cell belongs to which group. Okay, let's take 5 minutes break. We will come back to see how can we color those memberships into the visualization. Okay. Okay, let's continue. Have you got this step so far? Get this? Okay, good. So you have a C, and make sure C has the same number as the number of cells. So 2,509 here, you should have 2,509. Otherwise, there's something wrong with the computation. Okay, once we have that, there's a way you can color the scatters. Okay, so let's do this for 3D first. All you need to do is to put the C as a additional variable. So I leave a space here. This is indicate the size of your bubble. Okay, if you want to give 10, you can put a number. Otherwise, the default number will be internally giving. And the C just gave the color my cells members by the category. So in this case, you have the indication of your clusters and based on their membership, right? Okay, so you can do this for 2D plots as well. In this case, I'm going to try a larger. Just play with it. And then I see, okay, we'll do field those. And this gave you a picture like this. So some parameters you can make change to customize the presentation of your figure. Okay, next I'm going to use spectrum clustering to do the same thing and see whether we have a similar solution. So I will keep this still here, and I will generate another clustering results using spectrum clustering, and then I will generate this figure again and side by side and see whether they are similar. Okay, you're ready? The same type of code, it just replace this k-means using spectral cluster. Okay, you still get k, the same number of the cluster I want to make. And I will generate another c. So instead of using the same variable, maybe I can change to C2, okay? So in my workspace, I have a C and a C2, they are same size. It's supposed to be very similar. Now I will plot exactly the same kind of figure, but use, instead of C, I will use C2. Okay, see here, I just copied the previous code using C2. C2 is from my spectrum clustering. So what do you think? They generate similar results or not? On the screen, my left side is the result of the k-means cluster with k equals to 5. And on my right side is the result from the clustering, spectrum clustering. So certainly they look very different, especially when you have this group here, and then it's a small group. And then you have another small group right here. Okay, in this case, machine learning will not be able to help you further. You have to go back to biology manually, look at some marker genes in respective cluster the cells, because the information you gave to the algorithms, only the position of those cells, nothing else. Each cell in this plot, only the coordinates. The algorithm had no idea about each cell's profile of many other genes expression. It won't be able to tell you the difference between this cell and that cell too much. It's only based on the distance of their coordinates. In this case, only 2 numbers, maybe, or 3 numbers you gave to them. Do you have any other questions about this? Now I come back to this. I need one more output. Here, I'm going to export my expression matrix X. So I already have my S in there. Now I'm going to use original expression matrix to do my clustering. So I put X out. So my big X matrix contains information for 2,509 cells, but at each cell you have information for 3,850 genes expression information. So I'm going to use this to generate C3, another category output. Here I use run C3. So I need input is my big X. I need 5 outputs. So still my K is still the same. So you see it's a very similar structure, but in this case, well, you see immediately my computer report low system resource may affect the performance of my computer, because this algorithm, remember, will build distance matrix, will transform it, will chop in different dimensionality and do k-means. It will really take some time for me to finish. Then once we have a C3, I will visualize the membership of cells again and put 3 panels side by side. K means spectrum cluster and SC3. SC3 algorithm right now is blended from the S, the variable. You didn't give it S. So the algorithm doesn't know where you're embedding your cells supposed to be on the 3-dimensional or 2-dimensional space. It completely rely on the expression profile of the cells. So now I'm going to plot this out. I'm using C3. Okay. Okay. Just remind you, on my left is k-means results. In the middle, we have a spectral cluster. On your right side is SC3 algorithm generated. You see there are a few cells in the tip of this position here. In the t-SNE embedding, these 3 cells, for example, is grouped into this dark yellow cloud of cells. But SC3 consider them as either this group or that group according to the color of the membership color indicators. But overall, the consistent of those membership against k-means, it's actually highly consistent overall, right? Yep. By the way, this 1, 2, 3 different categories, the number, just categorical, so it doesn't matter your label is use what label. So 1, 2, 3 means nothing. It's just indicators. So that's why you see, okay, this may be number 1 category, but in this group has a number 1. Then they have different colors. Okay, are we all right, online students? So I'm going to move to another example. So I will let you to do it. Now you can clean all. So I'm going to load example data. So let's do, I need to change my directory, gene expression analysis. So I'm changing my directory into the package so I can load example data. Okay. Now the new data set I just load here is the one we used in the example, right? So now your job is to do clustering for this data set. Okay. And label them, use a color. So I'm going to use field. Easy to see. I try to cluster those cells, use k-means and also spectrum clustering. You can try SC3 if you have time. I will give you about 10 minutes to do this.
I can't access the example data in my installation, I guess.
Yeah, the first step, can you just search, see whether you have this folder?
I do, but I
do?
can't find this file in there.
It's empty.
Oh, I
Can
see
you-
it now.
Yeah, you can just click, go into this this folder, inside this folder, then you should be able to
Okay.
double click and you see it here, you can double click and load it.
Right. Okay.
Yeah. We just need these coordinates to play. Okay, on the screen, I finished these 3 different algorithms to do this clustering analysis. So I generate the C1, C2, and C3. So now I'm going to plot them on the same figure, use subplots, so they can put together 3 side by side. And first, I'm going to use figure. Instead of doing this individual figures, I'm going to use subplots. Let's see. So this will be C1. So I will create a figure and then do the subplot. This subplot will be 1 row, 3 columns. I will plot the first one here. So that's my k-means, right? I plot my second one is my C2. Subplot my third one, C3. Now I should have a figure which is not really good. Okay. So the first observation I can tell is k-means algorithm didn't respond to this data set very well, and because of the density of the cloud of points change greatly, and then you have 3 clusters based on their central node, and it doesn't look like what we expect to see, and the spectrum clustering and performs better and intuitively sound, and that's what we think this should be. However, if we look at the full spectrum, we consider the whole profiles of gene profiles of cells. SC3 just gave us something like this. There are some cells in blue, that's category 1, scattered around those astrocytes. And then another group and the yellow group. So the distribution is completely different from what we t-SNE embedding gave to you. So you don't see, I think you would see something like this if SC3 and t-SNE gave you quite different solutions, right? You actually don't see this very often if you have a high quality data. From what I have seen and experienced. Okay, so we are going to finish off with clustering analysis from here. Any other questions? In the online drive folder, I upload another example data set, which is in the zipped file called density classed underscore test zip. So in there, you have some other example data and also DB scan example code. So if you are interested in, you can download this zip file, unzip it in MATLAB folder, and then play with the examples. Now I just show you where it is, and then I will leave this for yourself. Okay, it's here. I use my mouse pointer to point to this file, and then it's a small file, but you unzip the couple of example code you can use. All right, so I'm now moving to another example to show you how to download multiple data sets and merge them into one. So the example I'm going to use, the geo accession number is inside this file. So I can double click and open this. This is a file which people collected mouse sample, and then they upload single cell data to geo database. And this study is mouse lung development, and they have a sample from a embryo 8.5 day, that's before the birth, and then 9.0, 9.5, 12, 15, 18, and also P0, P3. So there are multiple samples from a mouse lung, and then individually they process and then single cell RNA-seq. So the data set we need, for example, if we need 8.5 data set, we need to go to this geo accession number and go to this record. So I'm going to do yes, my computer is very slow right now. Let me go to the geo. Let me clean my desktop so I can make its computer faster. So now what I need to do. Yeah, all I need to do is this ID, right? So I need to remember this. GSE 13, well. So this is what I need. No. 6689. So I put this ID in the chat in case you want to copy. And with this, we can go to the page for this study with data. Okay. Okay, you see some of this explanation of the experiment protocols. And in this, you have publications, the link. And then we have 4 samples here. Oh, this is not a really good one because they already combine everything into this file. So let's use another example. Let's use this. GSE 165063. Okay, we are using this. It's also multiple samples from a mouse, but just different days. In this case, they didn't combine anything. They still have independent record for each sample. And for example, I want to download data from E12 and E15 and merge them into the same data set. So maybe I should try E15 and P3. So whatever, you know how to merge 2 and you know how to merge 3. And my computer right now is too slow. Maybe I only try to merge first 2, okay? So let's see, we still need MATLAB. I will need this ID. So that's our first sample will be this ID. GSM 502553. Okay. Then we need four as E15. So I will process them independently and then save them as two variables. So what I can do is to use this two and the C. Okay, I can get a C accession number. The first one. Oh, okay. Okay, so I'm automatically processing the first data. Okay, I have my first data set. I will do a quick clean up. So you know how to do the QC. I will make it really small, maybe with 500. 10%. Okay, I clean up my data, and then next you want to save this into workspace. So I will call this SCE1, single cell experiment number 1 sample, and then save it. Okay, so that's my number 1, and you can view the metadata that's from this sample created today. Now I finish this, I will try next one. So next one is this ID here, I copy. So I can close this. not saved because my information is already in SCE1. So I will repeat this to create my second data set. It was 43 and now it's 44. I think that also will take 1.5 minutes. How about your computer? You finish it? No, take long. Take me 4 minutes. So now I will do the same filtering, stringent, 10% expression. So after I clean this data, I'm going to save this into another variable. Okay, now it's clean. I use this export function again and export this current into a variable called SE2. Now in my workspace, I have 1 and 2, and I know they are comparable. I'm going to merge them. And the function is here in the experiment. And so now this, okay, that's a large data set, 10,000 cells. So I can see, okay, merge SCEs in workspace. So after I merge, I will load those merged and overwrite what I currently have here. So it's okay. So I just select yes. So I will select 2 variables to merge. So the first one, the second one, and then you have to decide, because they contain different number of cells, genes. Are you going to do intersection or you want to union? Intersection means you only keep the gene that appeared in both data sets. If you want to union, then the gene uniquely expressed in one sample will still be there, and another will be filled in with zeros. Okay. In this case, I just intersection, easy to merge and merged. Now you see you have 16,000, almost 17,000 cells. That's a lot. And they color coded in by the different batches. And then the merged, the first one is from that record, and that's from this one. So it's already merged. And then I can do Disney, whatever, the operation. And now you operate working on this merged data set. You can do this for 3 or 4 or more data set put together, because you really want to do this classification, clustering, and found the cell types as a whole group. Although they from a different development time, you want to do this systemically, and then you work out what's change follow the development stage, right? Okay, so I think that's all for today. And once I finish this, I will save. So next time, maybe we can play with the merged data set. You have 2 development stage cells to compare with, compare each other, right? Okay, that's it for today. And please, if you have any other questions before we finish, please ask. Let me know. Okay, otherwise, see you next week. Have a good weekend, everyone.